Yes, thank you very much for joining these workshops. And we have a series of them, as Jeet mentioned. And what I'm going to talk about today um, fits in really well with what Mike was talking about. My talk will be more on the bioinformatics side because it's a workshop where I will talk a little bit about knowledge bases and also analysis and how one can use different resources because one resource cannot do it all. So sometimes one needs to move from one resource to another to answer a scientific question or a biological question. So uh, with that, I want to say a little bit about where I am located. So I am here at George Washington University, which is right outside the Foggy Bottom Metro in Washington, D.C., walking distance from the White House and the National Mall and other uh, nice areas for tourist attractions. This is the entrance to the building that my office is in. And it's 15 minutes or 10 minutes walking distance to Kennedy Center. And also, this is the area where there are a lot of office buildings in this area, also World Bank and other offices. The people who we work with. So we have several people in the group. Some of them work on GlyGen, and there are others who work on GlyGen and some other related projects. We also have collaborators, international collaborators that was mentioned earlier from Japan, Kyoko, and then Maria from EBI and Preeti. Evan is at NCBI, Frederic is in Switzerland, and Paul is also at Bethesda, NCBI, NIH. Our advisory board is Lynn, Michael, and Rebecca. And I also want to acknowledge there are some slides I got from Darren Natali, who's not on this list right now. And then Chinwaki also, who works with us, some biocompute slides. So I will talk a little bit about the history, how GlyGen and some of the things that we're talking about from the glycoinformatics space, at least at NIH, some of the things that has helped us get where we are. There was a 2012 report that was published, a roadmap for the future, transforming glycoscience. And that kind of was a culmination of a lot of work over many, many years. And that report was really helpful in building some of the infrastructure that you're seeing around NIH and also NIH-supported research. So the glycoscience program in 2014 helped fund these two, or they published these opportunities, which we applied for, and we were fortunate to receive support from NIH in 2017. After 2017, we are funded through another NIH mechanism, and we worked, William, Michael, and I worked. William York, he has retired right now, but he very much helped start some of the things that you'll be seeing today. And then in 2020, Mike mentioned we got together and started the GlySpace Alliance because there are different resources around the world who are working towards bringing glycoinformatics to the forefront. So GlyGen and from Frederic, GlyConnect and GlyCosmos and GlyToucan and Kyoko, we all joined together and formed the alliance, which allowed us to share data, but also provide data with the CC 4.0 license, which means you can do anything you want there. You can download it, build your own resource, write papers. The CC 4.0 means the citation is expected, which helps us, of course, to make sure that we are serving the community. So today at this workshop, I will talk about or use 3 proteins as examples. These are the 3 proteins. One is a phosphodiesterase. The other one is interferon gamma receptor, and then EGFR is the third one. And I'm going to use these proteins as different examples because when glycans So one of them I'll talk about studying proteins from evolutionary viewpoint, and the other two I'll talk about, when glycans are attached to them, how to explore them and what can it tell us about their function. And before I get into all of that, I mean, there was one slide from Mike where he talked about how glycans are there in all domains of life. And I want to talk about the perspective from the evolutionary perspective. And I'm not going to only talk about glycans. I'm going to talk about proteins quite a bit because, the glycans that we are presenting today in this workshop are mostly about glycans, either attached to protein, or most of them are attached to proteins, and how we are capturing that information in GlyGen. So the tree of life, from 384 BC, Aristotle, he classified all living organisms as either plant or animal. And then Whitaker summarized them into 5 kingdoms: animals, plants, fungi, protists, and bacteria. 65, around that same time, the whole idea about using sequences to relate organism was being put forward by Zuckerland and several others. And there was a phenomenal pieces of work that was done by Carl Woos and others in around 1990s, which brought archaea to the forefront. So this is a very well-known figure where we have the eukarya, bacteria, and the archaea. And the discovery of archaea was almost like finding aliens amongst us, right? I mean, we didn't know that there was a completely different domain of life that existed before that. So, and all of these organisms that you see, of course, they have glycans on them. So when you're looking at the research space and the knowledge space in the bioinformatics world, I have to talk about Dr. Margaret Dayhoff. I mean, if you know her already, that's great. But if you don't know, just a little bit of background information. So she was a physical chemist and a bioinformatics pioneer. He was interested in the possibility of deducing evolutionary connections of the biological world, because anything that we see in biology, to better understand its function, one has to look from the evolutionary perspective. So she deduced the substitution matrix, the PAM matrix, MDM, or the Dayhoff matrix. And so when you look at amino acid and you look at the single letter code for amino acid, so she's the one who was doing alignments by hand, right? Trying to figure things out initially. And not having a single letter code, you can very well imagine, is really hard to look at alignments. And this was the first database, 1965. It was published as a book. And in one of these books, she talked about the single letter code for amino acids. And I think, some of the other editors in that book, they were not exactly thrilled about the idea, but they saw the benefit of it. So from that book came a lot of biological knowledge bases, biology knowledge bases, protein information resource, and then also UniProt and others. So you see these matrices in BLAST. So I have another slide on BLAST. So BLAST is one of the most widely used softwares in bioinformatics, right? And when it is trying to find homology or homologous sequences, they use a matrix like this. And what a matrix does is that what Margaret Dayhoff did, and others after her also refined it a lot, is they would look at alignments from closely related or distantly related proteins, and then they would try to understand how many of the amino acids are different between the two pairwise alignments, or it could be a multiple sequence alignment. And what is replacing, which amino acid is replacing what from an evolutionary context. So then they could have a score. They would put a negative score, likely to happen, or a positive score when there's a conservative substitution. Or identical matches were, of course, given high numbers. So using this type of matrix, you can then generate alignments, identify homologs, orthologs, and so on. So Janet Thornton, she recently retired. She and others, they have some amazing publications, and this publication, actually, a mutation data matrix for transmembrane proteins, that came out in 1994 is a great read to understand how people do these kind of analysis. So BLAST, as I mentioned, there's a nucleotide BLAST, there's a BLAST-X, T-BLAST-N, and protein BLAST. So DNA to DNA nucleotide BLAST, and protein you translate it to nucleotide on the fly, and you can do that to identify homologous sequences. And BLAST-X is translated nucleotide to protein, and then protein BLAST is where input is also protein, and the pairwise alignment that you're getting is also protein. And of course, protein BLAST is still one of the most used methods, and in terms of, or even nucleotide BLAST is still considered the gold standard in alignments in looking for homologous sequences. So even though there are lots of other aligners, including short read aligners and so on, you would end up sometimes just running BLAST on small number. It's very computationally heavy, so you can't do it for millions and millions of reads, for example. You still use it to better understand, are they indeed homologous or not. So this is a screenshot of BLAST result from NCBI, and this screenshot was taken several years ago. And why I can't do these type of screenshot as easily now is because the number of sequences in the databases have grown so much that if you just run BLAST at NCBI, you will find everything red, right? Because you will have thousands of E. coli or thousands and thousands of eukaryotic organisms very closely related. So you cannot see the differentiation. But early on, you can still do it. You have to create your own personalized BLAST database to do it. But this figure actually shows you can make discoveries, actually, or people make discoveries just using BLAST. So here you can see that this protein, this phosphodiesterase, has two domains. So in some cases, the domain is on its own. In some organisms, in other cases, they are two domains fused together. So you can actually also tell, just by looking at this figure, which regions of the domain is evolving at a faster rate, slower rate. So BLAST has been used quite a lot in the past. It's getting harder now, but to do these type of quick look at my protein from an evolutionary standpoint. Now, when you are trying to understand, and even for glycosylation sites, for example, there are quite a few publications on how conserved they are, because conservation most of the time equates to functional importance. So you can take a protein, let's say from human, and run a BLAST, and you have a plant and a fly and a worm and a yeast, homologs. What does it mean? What it means is that just by looking at the results of your BLAST, you can then say, oh, my protein most probably is 1.7 billion years old because the common ancestor has to have it. I mean, that's the most reasonable interpretation when you have homologs in multiple organisms. If you have homologs in all of the organisms, then it's 3.5 billion years old. And if it is that, that means that the gene has been conserved for a long, long time. And you can also look at the conservation of sites. Like if a particular position is conserved, then most probably it is important. And you can start asking these questions. How long has this site been there? So for N-link glycosylation, for example, if that asparagine is conserved, then you can start making this reduction. Okay, or maybe it might not be just the glycan is important, the site in itself is important, and that's how you try to understand what are the functional impact of that site if it is mutated. And that's what I'm going to talk about in my other slides, like a use case, a scientific use case, or that we can run through biological knowledge bases. But earlier, I mentioned the concept of domains and full-length proteins. So I think of domains as Lego pieces. So you can have multiple protein domains, and then you can mix and match, and then you can, create a dinosaur or a shuttle or a car, racing car. So that's what it is. So domain A can be with domain B in a particular organism, such as human, but in bacteria, domain A and B can be completely different proteins, or the B and A can be flipped in terms of where it is on the protein, or there could be insertions and so on. So all of this, what it does is that it increases the protein's functional space, the repertoire of things it can do. Now, from a bioinformatics workshop perspective, I want to talk a little bit about a few things which is might be new to some of you, but might be slightly, you may have a slightly different view of it. But I want to say what we think about certain things from the biocuration community. So what is annotation? So annotation, it's the same thing as a note in a way of a comment or explanation of something. So many of these resources have annotations. Sequence databases have sequences, but the sequence in itself doesn't mean much, right? A protein sequence in itself is not going to be of much help for you to understand what it does. You need to have annotations on it. Which positions are glycosylated, for example, which regions are phosphorylated, what are the interacting partners of this protein, because any wet lab experiment cannot be just done in isolation of knowledge of just one aspect, and especially in today's era where there is lots and lots of omics data and other types of information that can be attached to these proteins. It's important to have the full picture of the protein, what it does, where is it in the cell, where is it in which tissue they are expressed in, and a variety of other things. So that's all of that is part of the annotation that happens in many of the databases and knowledge bases. Now, the problem with it is that where is the annotation coming from? So things are known. Well, if there is a paper about that particular protein and somebody did the experiments, then it's known. So there is no argument. So somebody has to read the paper and say, okay, this is what is known about that protein. This is where the glycosylation sites are. This is the glycan I observed under these conditions. Then there are the obvious ones, the confident predictions with caveats, close similarity to experimentally characterized proteins. For PTMs is a little bit harder, just because two proteins are very close to each other in terms of identity, does not mean that the PTMs are conserved, or there might be heterogeneity, as also Mike mentioned. So it becomes a little bit more complicated in doing these type of predictions for PTMs. Then there are non-obvious, general or tentative prediction. So this happens when there are distant similarity to experimentally characterized proteins and the association-based methods. One has to do other type of bioinformatic analysis, gene or domain neighbors. You can do sequence analysis. You can look for motifs, functional motifs within the protein. And then there is unknown, that nothing is known about the protein. Nobody knows what it does. And it might be conserved, but somebody has to do the hard work, the experiments to figure out what it does. And that's where we are. There's still quite a few proteins that we do not know what it does, quite a few things that we do not know about the proteins, even though we have the basic function sometimes. Now, when we say we do not know anything about something, that is where research starts, right? But first, one must exhaust all the resources. And that is the key. If you are coming in from a wet lab, if you start working on a protein or a gene, exhausting all the resources is step one before the first experiment is designed. So that's where UniProt comes in, RefC, GlyGen, and other resources come in. Then there are non-obvious cases where the resources may not have the information, but you can use, and this part is where bioinformatic tools come into place. The user with some knowledge of bioinformatics can use tools such as SciBlast, HMM, association analysis, structure comparison, and other things to guess what is going on and predict. Then there are obvious cases of which looks obvious, but you want to avoid propagation of errors due to wrongly annotated proteins. Then there are knowns, which we talked about earlier. So misinterpreted experimental results, happens. And here's an example. There's a mistaken identity. So for if COB, so there is a gene name, let's say, which is used for two completely different proteins. So sometimes that happens. Is getting better over the years, though, because a lot of automated methods of annotation and identifying these type of errors has become easier. So from annotating a protein and getting information you can do BLAST, PFAM analysis. If it is not sufficient, literature pertinent to BLAST hits, you can start looking at the papers that the BLAST results produce. And then the advanced ones is you start looking evolutionary analysis. You generate phylogenetic trees, try to look and see at what in each of the nodes, what is the function that you can assign to the nodes, and any children underneath that node, you can say, okay, this is the function most probably these set of proteins have. There is direct biochemical characterization. If identical to a SwissProt entry, let's say, which is manually curated in UniProt, then you can use that. If it is similar to existing SwissProt entries, then you're in luck. You can transfer some of that information. And then if there's strong similarity over the entire protein, you can say, okay, this protein must have this function. But sometimes if it is only strong similarity over just the domain in a protein, it's not as easy to transfer functional information. So here's an example that I want to run through. So all these dots you see here are different proteins. And if you run it's protein, they're in the space where based on the identity, based on BLAST hits. So let's say these are the proteins, a group of proteins that you have, and you can find papers for this protein. So the one in the stars, one is acid kinase, one is a protein kinase, one is a glycerokinase, one is a ribokinase, glucokinase, and hexokinase. So if you find you can then start looking at relationships. Okay, so this smaller circle inside, they all are ribokinase, and the distance similarity or distance between them are very small and similar. So any protein in that group is a ribokinase. And then you have other ones still there. So if you have a protein kinase, and then there is a protein which has similarity to a protein kinase and a glycerokinase, then and a ribokinase, but they are equidistant from all of them, then it becomes a little bit trickier. So you can start saying that, okay, so at the bigger level, they're all kinases, and then the next level down are sugar kinases, and then you have the ribokinases. And if you have a protein which falls there, it's a sugar kinase. So that's how one would look at functions and try to assign functions in an automated fashion. And at some level, though, sometimes just identity across the entire sequence is not enough. And that's where the concept of site, a site in a protein, that's really important, including the PTM sites. So in this particular example, there are these two sites, the D and the K, and based on the similarity of this protein, based on this tree, you can say, okay, these are inorganic phosphate binding phosphofructokinases, these are ATP binding phosphofructokinases, and these are also inorganic binding phosphofructokinases. But then, and all of this you can say because within that family, there is at least one characterized members. Somebody wrote a paper, characterized that protein. But in some cases where you do not know, or there is no paper within that family which tells what exactly it does, it might be a little bit risky to pass on that annotation for that protein. So you can build rules to do this. So you can say, if I have a protein and I want to know if it is an ATP binding or inorganic phosphate binding, then depending on whichever it is close to, then you can say, oh, this protein is close to ATP binding, then I can call it ATP binding phosphofructokinase. But then if you do not have close similarity to any of them, that's when you can say, okay, this is just a plain phosphofructokinase. So that was a brief background on how to look at protein sequences. And I'm going to now talk about the use case that I mentioned earlier. How do we use we have to come up with a question. So this was a paper written by Rolf Apweiler and others on frequency of protein glycosylation as deduced from analysis of the SwissProt database. This is a paper from 1999, so 23 years ago. Or 24 years ago. So it was quite some time back, but this paper still holds some of the basic things that they were talking about. In that paper, they say the SwissProt protein sequence data bank contains 75,000 entries, almost 2/3 of which include this motif for N-glycosylation, NX, S, or T, and thus maybe they could be glycoproteins. So we conclude that, and based on that paper, they conclude that majority of the sequence containing proteins will be found to be glycosylated, that more than half of all the proteins are glycoproteins. And this also emphasizes something that Mike was talking about. Now I'm going to present to you two really interesting papers. One, actually more than two. I have a few papers that I want to present to you. So I want you to remember about what we talked about glycoproteins. I want to remember what I talked about sites. There has to be an aspargine for it to be N-glycosylated, right?
Sorry, Raghav. Sorry to disturb you. There's a meeting reminder on your screen, so if you would want to make it go away.
All right, thank you. There were three. So this is a paper by Vogt et al. In 2005 Nature Genetics. 3 children have the same missense mutation in the same gene, which gives rise to this inherited form of deficiency due to the expression of some receptor on the cell. So the missense mutation, in this case, the missense mutation creates a gain of glycosylation site. I mean, it's called consensus site, but basically there's a gain of glycosylation. So this paper was published in 2005, and it was actually one of the papers that I saw around that time and started thinking, wow, so the gain of glycosylation is something that really is interesting. There were other papers. There's a more recent paper in 2020, which confirms what they found in 2005. Mendelian susceptibility to mycobacterial disease is a rare symptom that confers predisposition to illness caused by moderately virulent mycobacterial species, such as the BCG vaccine. The BCG vaccine has a Mycobacterium bovis, I think so. And non-tuberculosis environmental mycobacteria by the more virulent mycobacterial tuberculosis. So what it's saying is that gain of glycosylation was confirmed. It does do that. So this is the paper, and from that paper, they show this table from that paper that they show 8GMD, by the way, is a database of mutations found in humans. And what they found is that in that paper, there were these many mutations, missense mutations, and then out of them, these many resulted in gain of glycosylation mutation. And then these many resulted in loss of glycosylation mutation. So since then, 2005, there's a lot of other mutations that have been discovered. So this analysis, if you do it today, you will get larger numbers. Of course, these numbers will stay, but you will get. So what it's trying to say is that when we go to dbSNP or GWAS studies and other, the GWAS catalog, I mean, and other places, you will find a lot of mutations that are there, like polymorphisms are there, mutations are there, and many of them could result in loss of glycosylation or gain of glycosylation. And fast forward, this is a great paper in 2019. And while I was preparing one of my talks, actually, I came across this paper because I'd seen that paper in 2005. I went to GlyGen and I started, I wanted to find the protein just to see. And in GlyGen, I ended up on the table for this protein. The URL is there and the slides will be shared. So you can take a look at that URL. So when I go to that URL, to that protein entry, there is a row in the table. And this is the row that the 2005 paper, Nature Genetics paper is talking about. 168, in position 168, there is T2N mutation. And then, by the way, this is all data collected from different places, right? So what we do in GlyGen, or what people do in UniProt, or people will do in other knowledge bases, we collect information from a variety of places. And that collection results in information which is a little bit harder to track down, not little, quite hard to track down, because if they're in different places. So when I look at this, I see there are 2 publications that were associated. And one of the publication is this publication, which pretty much from a structural viewpoint shows what exactly is happening in these patients. So what they show here is that the structure, it's T2N directly at the site, directly in the phase, it causes a steric bulk that would result from the addition of the N-glycan and would introduce steric clashes, likely to prevent the interferon gamma receptor from docking to the high affinity intermediate complex, the interferon gamma and the R1 complex. So this is what we expect to see when we are trying to see. So let's say between 2005, what I want to emphasize here, if between 2005 until this paper came out, if you were to find similar examples, and if you have interest in structural biology, understanding the role of glycans, these are the type of experiments that knowledge bases can help design. So that's what I want to emphasize in this workshop. And then there's another example. There are other examples, by the way. So this is an example where a gain of glycosylation, which from a experimentally how you can prove. You don't have to always do structural analysis. So in this particular case, the gain of glycosylation, when it is there, they introduce this mutation in a model organism, and they start seeing in 15 days the tremors. So with the gain of glycosylation, it is a phenotype of this particular disease. The mice starts getting tremors, right? So the 15-day, 1 month, 6 month, you can see what's happening till the die. So this gain of glycosylation is attributed to that phenotype. So there are many such discoveries that are waiting to happen. This is another figure which shows the rotarod analysis, where actually the mice they go on this rotating equipment, and if they don't have the muscular, they cannot hold on to it. They fall gently down, so that and that's calculated to see if they are losing any of the motor functions. So this gives you an idea about how discoveries can be made by looking at gain and loss of glycosylation, of course, amongst other things. But I just wanted to show one use case here. To continue on that, how does it look like in GlyGen? How do we present this information? So these are screenshots from GlyGen, and you have the sequence here. There are many ways to look at the sequence, by the way. This is one way. And in that particular sequence, and this particular N, this glycan is attached. And then if you look more, click on some of the additional pages, you can end up looking at the glycan. On the right-hand side, the same glycan, and then you can select, like click on hexose, let's say. As soon as you click on hexose, all the hexoses will light up, and everything else will be kind of grayed out. So you can look at the sialic acid, you can look at the fucose, and you can explore your glycan structure within the interface to better understand what's going on. You can also go and identify which enzymes are responsible for putting those glycans there. And then you can explore those enzymes within GlyGen, or then from GlyGen, you can go to UniProt, or you can go to RefSeq and other resources. So coming back to looking at a large scale analysis for these type of things, I want to spend a little time on this figure to give you an idea how one can do interesting things. So you can get functional site information. That means protein sites which has functional information. So you can go to what is called UniProt KB FT lines. The line type doesn't show on the web interface, but if you go up and click on download and text, in the text version, it will tell you these are feature lines. So they have active site, catalytic active site, binding site, modification site. RefSeq CDD is another project that has been going on for more than 2 decades, to the best of my knowledge. They take protein structures and they look at the structure. The curators will look at structures and they will annotate the active site in the protein structure. And you can visualize all of this in CN3D. Jeet, in one of the workshops later on this month, will show how one can explore those sites, like particular positions in the protein in a 3D structure. Then there is DBPTM, and then other resources which will also have information about particular site-specific information. So on the left-hand side is functional site-specific information, and on the right-hand side is cosmic, all the places where there is single nucleotide variation data that we collected. So you can collect data on variation, collect data on sites, and then ask the question, how can these variations, what is changing in the protein function? So most of the current pathogenic mutation software that you have, what it will do is that it will look at an algorithm which is looking at conservation score and trying to say, okay, how pathogenic it is. So this is much more sensitive way of looking at it. You are not really seeing it, telling it's pathogenic. You're going a step further. You can say, oh, it's pathogenic, and it's pathogenic because it knocked off this glycosylation site, and now where there used to be a glycan attachment place, it's not there. So using these type of approaches, you can actually start making profiles, and you can also ask simple questions, which these mutations, is it resulting in a gain of glycosylation or a loss of glycosylation site? And in the sequon, is it in position 1 that you see a lot of mutation, position 2, which is the X, remember, from the motif, or position 3, which is the serine or threonine? So you can slowly start understanding what is going on within, even at the motif level. And once you have that, you can take all the PTMs and you can do cluster analysis and ask questions like, are there certain cancers, such as breast cancer or lung cancer, where certain type of PTM loss is happening a lot? So you can see from this figure that it is looking like that there are certain post-translational modifications that are being impacted more by certain types of cancer. And when you do these things, you better meta-analysis, you can also start identifying certain cancers and proteins, and which proteins map to which cancers. And you can say, okay, what are the biomarkers that you can get out of these type of analysis? So this is something that we already published, and you can take a look. But others have also done similar work, since we also we have been doing similar things. And our goal is that to allow these type of extraction of data directly from GlyGen and other sources that we will collaborate with. You can look at it from the drug perspective. So when there is a mutation which is resulting in some modification within the protein structure, or a gain or loss of glycosylation, is it affecting any drug binding site? So there are a variety of ways. So now, changing gears a little bit. So this is from Dr. Kasei's paper from GlyGen. So we look at GlyGen in this frame or data model. So we have protein, we have glycan, and we have a glycoprotein, which has a site, right? So this is, and then there's glycosylation that's happening. And from the protein, you can connect the protein to biomarkers, variation, phosphorylation, disease, and a whole variety of things. From the glycan, you can also connect it to a bunch of things. And when you connect everything, you can build knowledge graphs or do queries using Sparkle, for example, to querying our database. Now, what do we have in GlyGen? So we have Drosophila, we have yeast, we have Dictyostelium, human, mouse, rat. We have some synthesized glycans, HCV1A, 1B, SARS-CoV-1, and CoV-2. So we don't put every proteome. So we bring in the proteome first, because we believe that when we are trying to look for connections, you need the entire proteome. And then we also bring in the glycans, all of the known glycans associated with that organism, into the resource. So we can also look at the enzymes that are involved in synthesizing the glycans. You can look at other things. So the ability to look at it from a more systems level is what our hope is, our goal is. How do you find something? Well, there are, when I teach a class or do something similar to this, I say, use Google or Yahoo or whatever you use, Bing, and you can say GlyGen and then the gene name, EGFR. Usually the first hit will be the GlyGen page, right? Or, of course, you can go to the website, glygen.org, and type in EGFR. And so the first way that I showed is that you are going in, entering the resource using a gene name, a gene symbol. There's another way to find out something about your protein. You can do BLAST. So you can paste your amino acid sequence here in this field and hit submit, and you will get results. So our BLAST results is a little bit different than what you might have seen before. We also have ways to look at the text version of the BLAST results. You can see on the right-hand side, you can select, after you get the results, you can select, show me all the N-linked sites, or you can show me all the sequence. So one thing that in this particular example, I want to show that you can look at the sequence that are present in the subject, and you can see if the subject and the query sequon is conserved between human and mouse. That again, from an evolutionary viewpoint, you might start asking questions. Okay, if they're conserved, do they have similar glycans under similar conditions? And if there are functional knowledge that is present in the model organism that can be transferred to human, or the other way around. So it's a really powerful way to also explore the resource where when you start coming in at the sequence level. And then we have implemented some tools which allow you to do or get results like some of the figures I showed earlier. So I can go to the site search and I can ask questions, show me all proteins, which has a glycosylation site and a single nucleotide variation and mutagenesis. Mutagenesis data is where somebody took that protein and then they mutated it and to understand its function. So there is functional information that happens or one can get when they mutate that particular amino acid. So this will give you a list of proteins, which is a loss of glycosylation, right? Which will tell you what's going on. And some of those proteins, when you find it, you will find that other people have already published papers on talking about that loss, or you will find nobody else has done any work on it yet. And that's where maybe new avenues for research will open up. So the results, look like this. From that, on the left-hand side, you have a way to filter it, filter the results. The filtering is very similar to many of the travel resources, right? You search for a flight, and then it will tell you, do you want to non-stop or one stop or whatever, or hotels and things like that. So you can filter through. So instead of trying to make a very complex query up front, we want to be somewhere in the middle. You start with something, and then you can filter it till you get what you want. In this particular case, there's clusterin, where there is a single nucleotide variation that has been mapped to a glycosylation site, and also there is mutagenesis result, which makes it a really nice target for my use case, where I want to see, let's say, what is going on, what is the functional impact of the glycan site being lost, glycosylation site being lost. And this is a similar to a knowledge graph, but concept-wise is telling you what do we have. It's dynamic. So we have 10 organisms with these many genes and these many diseases are mapped. We have the protein. These proteins have glycans. This protein also have sites, and those sites may have glycan information on them. And there are enzymes which synthesizes the glycan. And then these glycans have functional motifs that are well defined by the community that Dr. Nathan Edwards has been working on. So all of this gives you a bird's eye view of what is there. And you can click on these different boxes or different search images or these lenses, and you can get results like the previous table that I showed you earlier. So when you click on one of them, you will get a result like this. So you will have a super search result. And from that result, you can see how many of them are in N-link complex, high mannose, hybrid, and so on. And by amino acid, how many of these amino acid sites are this functional information is attached to. So this is, again, a very unique and a powerful way to explore the resource to identify potential experimental targets. So coming back to that example, the EGFR example and loss of glycosylation. So when I look at the entry, the EGFR entry on the sequence page, I have a similar box on the right-hand side. By the way, you can also click on ProtVista, which is a product from EBI, European Bioinformatics Institute. We collaborate with them, and it's also integrated within GlyGen. You can have a nice graphical image. Here you have, if I click on N-link site, okay, so I have all the N-link sites. Then I want to know which of those sites had mutation. So it turns out that there is a mutation information here for this particular position 361 for them. So I go down, scroll down to the table. So the entire motif is NCT, right? So that's the N-glycosylation motif. And we see that in somatic mutation, there is a somatic mutation in brain cancer, where in 63, there is a mutation, not necessarily on the asparagine, but in that motif. T2I, there's a point mutation. So this is the kind of information that one can look. And then also there is a germline mutation, which means there's a population. So not only there is somatic mutation in some cancers, but there's a population where there's a germline mutation also present in this. So you can click on it and you can get the RSID, the dbSNP ID, and explore. You can explore the papers associated with it and a variety of other things. So this is how one can explore the different sites. You can also do gain of glycosylation. So in this particular case, 654, there is a gain of glycosylation NNI to NT, and again, due to point mutation. So having said that, some of you are informatician, so you might be interested in getting the data and writing your own scripts. So there will be some discussion about that in the workshops later on. So GlyGen portal will give you the information that I showed you so far. There's a Sparkle endpoint. We also have a portal which is we organize any data that we get. So let's say if a collaborator, including one of you, have a paper and you have a table of glycosylation sites and the glycans attached to the sites in a certain condition or a certain experiment. So if you were to share the table through GlyGen, then of course, your paper will be cited, the name gets cited, and that table stands on its own. And there is a README file associated with the table, which says how the data was brought into GlyGen and what are the assertions for that table, what is the evidence. And we use biocompute objects, which explains exactly how the data was processed, which allows others to reuse it. So there's a whole thing about reusing of data you might have seen in publications and communiques from NIH and other funding agencies. There is another project that we are very excited about. There was a paper that Jeet and others published on glycan structure dictionary. So the issue with the difference between, so in genes, recent papers, for example, the majority of the papers are using the gene names that are HGNC gene names, which means that there's a standard that most people are following. But when people are describing glycan structures, there is quite a bit of variability. So this is step one, we believe, in text mining, and also maybe in the future when people are writing papers, they will use the structure dictionary to pick up the term that is widely used, which will help in AI, machine learning, and other kind of things that we are not talking about right now, but we have some people in our group interested in. So take a look at this glycan structure dictionary. And in terms of the data set, we have close to 700 data sets, and there's a JSON format for those of you who are interested in that. You can parse it. There's a human readable README also, and then of course the downloads. There are a couple of other things that I wanted to just touch on, is that we are part of this common fund data ecosystem, and we are collaborating with all of these other groups. And it's a really, really amazing way to see how our entities, the gene, the protein, the glycan, can be mapped to information that has been produced by so many other projects. So this goes over and beyond what you can see or find, let's say, in GlyGen and UniProt or GraphSeq or any of the places, right? So it will tell you in Kids First or GTEx or Human Microbiome Project or IDG, what eliminating the druggable genome, what additional information is there for my gene. So for example, this is a quick demo of, like, if I go to the web page for Common Fund, go to vocabulary, go to gene, click on gene, I get I can type in EGFR, and here I can see differential expression of EGFR in human signatures. I can also see EGFR is connected to which glycan. I can also do other types of analysis from other resources, which again is allowing a user to find more information about the gene or the protein of their interest, to identify maybe some trends or some interesting items, which will allow them to better understand the gene or protein. There is also workflows one can do, part of the Common Fund, one of the partnership, where you can start with one analysis type, and this is again more for bioinformatics folks. They can start with one, you can input other pieces of things in there, and then you can build an entire workflow to answer your biological question using the Common Fund Data Ecosystem Playbook. Here's an example of a knowledge graph also. That's another project where you can take different proteins and you can use knowledge graphs to find relationships that you wouldn't find just by doing a Boolean search. So here is another example how one can use the different data sets to do that. So if you want to share data, this is the link where people can upload their table, or you can just contact us. Some of you may have already seen the data management and sharing policy that NIH has been sending out, and I'm sure similar things are there being sent out in Europe, Asia, South America, and other places. There is a big push to share data, and we hope to make it easier for people to share data, or if GlyGen is not one of them resources, we definitely feel that we might be able to point you to other places where your data can be better shared. Get in touch. This is the URL, first name, last name, subject, and let us know. There's a plugin for a biocompute conference that we are planning on having, which will more for bioinformatics folks in FDA, where defining how a workshop needs to be documented for it to be reusable. With that, thank you.